So welcome to that next class. So in that class we will discuss the blood composition of blood composition. So you can see in that figure. Okay. So you can see in that figure their blood. Previous day we have discussed the plasma and blood cells are there in the blood. So today first we will discuss the plasma section. So the compositions of the plasma. So you can see in that figure there is a plasma, and this plasma contains the water, and it's about the 91.5%. It's a watery substances. Another things is the proteins. So proteins are there. So you can write here. There's a in if I write down plasma, it's the major component is the water. It's I have just written down the H2O. So it's about the 91.5%. So remaining percentage are the it's like if you think the other things in that plasma, it will be like the organic portions. It will be like protein. So proteins are important. Which type of proteins are there? You can see in that figure. It's a albumin is there, globulin is there, and fibrinogen are there. So other solutes also presents. It's like electrolytes, nutrients, gases, wastes, and regulatory substances. All are there. In all are about the other solutes. It's about the 1.5%, and proteins about the 7%. It's all about the nearby. So proteins are there. Major is the water substances, proteins, and the other solutes are there. Now we will go through the protein. Which type of proteins are there? So its protein, the major protein is the albumin. Albumin is the most abundant protein. Albumin. So first of all, most abundant. Most abundant protein in the blood, that is a plasma protein in the blood, that is the albumin. So what are the major functions of the albumin? So maintaining osmolarity. So it maintains the osmotic pressure and transport. So main function, if function is that maintaining osmolarity. Osmolarity. Maintaining the osmolarity. So that is the major function. If so, that means it holds the water. So in another sense you can write down, it can hold the H2O. H2O holding, it's completely depend on the albumin protein. So what happened if it is not holded the protein? So now we are discussing the albumin. In albumin section, maintaining the osmolarity, and that's why it maintains the blood volume. So blood volume maintain because it holds the water, and another role is the it acts as a buffering role. So if albumin is decreased, what happens? The plasma cannot hold the water. So what happened? Water going to the tissue level. So water loss at to the tissue level. In marasmus and quasi-core disease that happened. So that time the protein deficiency or liver malfunctioning is happening, and that is called the dietary deficiency. So if albumin get decreased in plasma, that can cause the liver malfunctioning, that also the marasmus and quasi-core disease. So that is the albumin. Now we are going to the globulin. So in previous slide you can see that the globulin, it is all about 35 to 36% is there. So blood level is 2.0 to 3.5 gram per dl. So its main responsibility is that the immune function. So it's a immune response. It regulates the immune response. Which kind of molecule, that we will discuss. So generally four type of globulin are present. So which type of globulin? I'll just write down: alpha globulin, alpha, beta, gamma and delta. So these four type of globulin present in the plasma. Gamma globulin, that is responsible, that is also known as the immunoglobulin. That is the immunoglobulin, IgG. So it acts as a antibody. So that produce in the B cell the gamma globulin. In normal condition, amount of albumin is more than the globulin. So normally, in normal condition, two protein we have just study now. Albumin its amount is much higher than globulin. We'll going through that complete details later. Now during infection what happened? Because this is the antibody section, so that will increase the antibody in disease section. In disease condition, the globulin will be more than the albumin, and that time the albumin getting low. So what happened? So in disease condition, albumin and globulin. Now somehow we can check the different images, like we can draw some plot like that. So this is the concentration and the albumin. First we'll put the green section, that will be the albumin, that is the level of albumin, and other things are like in small. See, it will be the alpha globulin, then will be the beta globulin, and then will be the gamma globulin, because for the infection portion, and it will store, and it will be the delta globulin. So that is kind of you can say the protein map. But in the inflammation section, that's in normal, that is the in normal condition. If any person is in disease condition, it will be like, if in disease condition their first albumin portion getting low, it will be low, alpha, beta and the gamma will be more and delta. So in disease condition, gamma globulin will be more in concentration. Hope that everyone is understood. So we are just studying about the different protein. So now we have studying the albumin, globulin, and the another protein that is known as the fibrinogen. So fibrinogen, it is the protein that the 4 to 6%, that is mainly the clotting factors are present. So if in previous class I have discussed that the blood cell, if you eliminate the blood cell uh, from that complete blood, it will be like plasma, and if you remove out the fibrinogen part or the clotting factors, then it will kind of serum. So fibrinogen are the important key factor of the protein. Now we will discuss the other type of protein. Some of the enzymes are there in that plasma section, and some amino acids are also there, and lipid. Let's go. You can write down the notes also. So now let's take the different color blue. Okay, let's say what are the enzymes. Now we can say D is enzyme, albumin A, B is globulin, C will be the complement protein. I will discuss later. Complement protein, it means during the infection. Okay, let's discuss now. So in during the infection, that is the infectious time, during the infection, what happened, gamma globulin is more active, and in that condition they are inactive form of the complement protein that get also active. So these complement protein are also present in the system. So now I can see is the complement protein, complement protein. Okay, let's go for the C, complement protein, the part of the immune system. Albumin A, write down the A, and globulin B. Globulin is the B portion, and C is the complement protein, and D is the enzyme that are present in that plasma. So what are the enzymes? Enzyme generally, it you can say these are the clinical marker. Clinical marker. This marker, normally this marker are mainly checked during the disease condition. So what are the marker? I'll just write down amylase. Amylase concentration increase or decrease, or you can say lipase. Lipase or amylase condition when increased, that can indicate that the pancreatic disease. So this is a pancreatic disease by the amylase and lipase. Similarly, creatine kinase, if it is increased, that means problem in that heart, muscle or liver disease. In the lactate dehydrogenase, so what happened in lactate dehydrogenase? If it is increased from its base level, that indicate that heart and liver disease. Another is that alanine transaminase. Alanine transaminase. That is also key important factor for checking the heart condition. It is also responsible for the heart, liver diseases. And basically alanine transaminase is mainly responsible for the liver disease identification marker. We may also, their name is getting changed somehow, like that SGPT and SGOT. SGPT and SGOT. What is the SGPT and what is the SGOT? SGOT is that serum glutamate oxaloacetate transaminase. Serum glutamate oxaloacetate transaminase, and SGPT is a serum glutamic pyruvic transaminase. Serum glutamate pyruvic transaminase, serum glutamate oxaloacetate transaminase. Both are the checking by the in liver disease. So these are the key enzymes present in that plasma. So now we are discussing about the different components of the plasma. First we have discussed the albumin, second we have discussed the globulin in the normal condition along with the disease condition, and third we have discussed the complement protein. Fourth we have discussed the different kinds of enzymes present in the blood, and like amylase, creatine kinase, lactate dehydrogenase, alanine transaminase, and along with the SGPT and SGOT and their clinical condition, how they changes are, along with few of the amino acids are present in that blood, like the glutamine. It's most absorbent amino acid and the major transport form of this is alanine. Another thing, the lipids are also present. You can say give me E or F. The lipid, some lipid molecules are present like the TAG, free fatty acid, and the cholesterol. So these are the key components, and if you think about the nutrient, if I give the F nutrient is the glucose. The major fuel of the body is the glucose. So in organic sections of the plasma membrane we have discussed. Now we can give a brief things of the inorganic, like some of these cations and anions are present. Like cations, what are the cations present in that blood? Let's write down. Sodium is present, potassium, calcium 2 plus, manganese 2 plus, Fe plus plus or Fe 3 plus. All the forms are present in the plasma. So these are the cations, and anions, what are the anions? Like chloride ion Cl minus, SO4 2 minus, PO4 3 minus, bicarbonate ion. They are the major, bicarbonate are the major buffering system present in that blood. So now in this video lecture we have discussed the only compositions of the blood, compositions of the plasma. The aggregately we get more details in the disease condition and along with the normal condition what happened in our body. So in next class we will discuss the cellular component of the blood. We will discuss in the next class. Thank you.